then when I graduated college, I couldn't decide if I wanted to go to medical school or grad school. And so what I did was I decided to take a technician job, actually at Johns Hopkins when I was about 20 years old. And then I volunteered in the hospital. And so when the hospital where I volunteered, I worked with little kids who had cancer. And I quickly realized that I'm way too soft to do this as a profession because I got very attached to the kids, and then, one or two of them passed away, and I was like, I cannot do this on a daily basis. And so I realized my place was at the bench and not at the hospital bedside.
Hi. Welcome everyone to another interesting session in the series of interviews conducted by the Department of Chemistry, University of Colombo, to inspire us in the field of science and bringing up great insights from eminent scientists. I am Achinta Ranaganga, and today Lakshmi Sivalingam and Shyam Ranavir also join us as interviewers. We all are fourth-year undergraduate students following a special degree in biochemistry and molecular biology at the University of Colombo. Today, we will be interviewing Professor Ashi Weeraratna from Johns Hopkins University. I am proud to say that Professor Ashi Weeraratna is a Sri Lankan-American cancer researcher whose findings contribute to the scientific understanding of melanoma tumors. She is the Bloomberg Distinguished Professor of Cancer Biology and E.V. McCallum Professor and Chair of the Department of Biochemistry and Molecular Biology at the Johns Hopkins Bloomberg School of Public Health. She is a member of the National Cancer Advisory Board, which assists the directors of the National Cancer Institute in the activities of the National Cancer Program.
It's a great privilege for us to have you today. And thank you so much for sparing some valuable time of yours to take part in this interview. To begin, would you mind giving us a brief introduction about you and your family background?
Sure. Thank you so much for having me here. It really is such a pleasure to do this for my birthplace and to see the amazing talent that's coming out. That's wonderful. So I was born in Sri Lanka, but I left when I was 2 and I grew up in Africa. So my parents, my dad was actually one of the first Sri Lankan officers in the British Royal Air Force. And when he came back to Colombo after being stationed on the Isle of Man for many years, for many months, he decided that he was going to help to start one of the regiments of the Royal Ceylon Air Force. And he was just an amazing, formidable man. His name is Ashi Weeraratna. And he passed away very sadly in 2000. We had his funeral in Colombo. And, it was so overwhelming to me. Like, when you're a kid, you don't really realize sort of how distinguished your parents are until, you're an adult or something tragic happens. And so when my dad passed away, the Royal Air Force, the Sri Lankan Air Force turned out to give him, a salute and a processional. And I didn't realize really until then how much he had done to start, to sort of start the Air Force in Sri Lanka. So that was really fascinating. After his time with the Air Force, he was very interested in the economic development of developing countries. He worked with Ceylon Tobacco for some time and then decided that we would move to Africa because what he wanted to do in Africa was to help people, sort of deal with the after effects of colonization. So as you all know, when a country is colonized, you have people who come in from Britain or Portugal or France or wherever, and they set up all these systems, many of which are good for the country, but then they staff them with people from their own home countries, and then they leave and those systems break down. And so my dad's job was to help developing nations deal with that transition. And so he was very much a socialist and very, invested in the developing countries. And I think I learned a lot of my life philosophy from him. He would always say, if you give a man a fish, he'll eat for a day. But if you teach him how to fish, he will eat for a lifetime. And so I think that's something I have definitely taken away from my dad. Yeah, I grew up in Lesotho. I was there till I was about 17 and a half. And then I decided I wanted to go to America for college. And so I was fortunate enough to get a position. And I came to a tiny little college in the middle of, Southern Maryland, and only 1,200 people in the whole college. So it was very small, unlike some of the other colleges here where there's 50, 60,000 people. But it was such a lovely place, and I made a lot of wonderful friends. And I think one of the most for me, formative experiences at that college was that they gave internships. So in the summertime, you could go to somebody's lab and they would pay you to do that. And you could learn, valuable lab skills in some of the top labs in the country. And you could work to pick any lab that was to your interest, and the office would work with you. So I always thought that was really fascinating. And it was so helpful to me. And then when I graduated college, I couldn't decide if I wanted to go to medical school or grad school. And so what I did was I decided to take a technician job, actually at Johns Hopkins when I was about 20 years old. And then I volunteered in the hospital. And so when the hospital where I volunteered, I worked with little kids who had cancer. And I quickly realized that I'm way too soft to do this as a profession, because I got very attached to the kids. And then, one or two of them passed away. And I was like, I cannot do this on a daily basis. And so I realized my place was at the bench and not at the hospital bedside. And so I decided I would go get my PhD. And I went to George Washington University in DC, where I got my PhD. I came back to Hopkins very briefly for just a few months to do a short postdoc. And then I went to the NIH, where I stayed for sort of the next decade. And from there, I went to the Westar Institute, and now I'm back at Hopkins where I started. So a long circular journey.
Well, madam, Professor, prior to joining Johns Hopkins, what was your role at Westar Institute?
So when I started at Westar Institute, I was an assistant professor, sort of beginning my lab, but not really, because I had sort of run my own lab for a couple of years before that. And then when I started at Westar, it was really the first time I learned how to write grants and that sort of thing. And then eventually became a full professor there, and became one of the leaders of here we have a lot of institutes have a cancer center within them. And the cancer center has a structure where you have like a cancer center director, and then you have different programs. And so I led one of the programs on tumor metastasis and microenvironment.
Professor, what do you think is the most fascinating aspect of science for you?
Oh my goodness, there's so much. I feel like every time we look at something, we learn something new. I always tell my students the data are the data. You should never get married to a dogma, because you're going to have to disprove it sooner or later. So we just had a recent publication where, it kind of turned about 10 years worth of work that we thought we knew about a pathway on its head. But it was one of my favorite pieces of work that's come out of the lab precisely because it did that. And so I think for me, the most fascinating aspect of science is the more you know, the less you know. So, and what I mean by that is simply that, when you're researching a topic, it's so important to always keep an open mind, and, be open to the possibilities that you were wrong, and be open to the possibilities that, as you dig deeper, you may find more and more interesting things.
Professor, may I know how did you become interested in the study of cancers, and was there anyone who was influencing you to select this field?
Yeah, that's a great question. So I've always been very interested in biology in general from the time I was small. I remember when I was about 12, I had this research project in my school in Africa where they had sent us to the garden to dig up some dirt and then put the dirt in a dish with water and some grass. And then they said, okay, now you're going to look at that dish in the microscope every day and you're going to draw all the organisms that start to appear. And it was fascinating because, the first day it was just kind of like this dirty, muddy water. You couldn't really see anything. And then over time, as it got more and more pungent and disgusting. There was algae, diatoms, little amoeba growing in it. So I was so fascinated by that. And my sister, who's considerably older than I am, she would drive me on the weekends so I could go take pictures of my experiment. And every time we walked in, she'd say, "Oh my God, it smells so bad." And then she would always say to me, "Oh, I don't want to drive you to," she would drive me there and then say, "I'm not coming in because I don't want to smell your festering hay cultures." And so she would patiently wait outside for me. But so I've always loved biology. I've always found it fascinating. And then someone who was relatively close to me developed cancer and passed away. And that's when I became interested in the study of cancer and trying to understand the disease.
What was your most significant scientific discovery, Professor?
So I think for us, it has really been our last decade of research on really trying to understand the molecular level at which the process of aging regulates cancer. So about a decade ago, maybe even a little more than that now, I worked at the National Institute on Aging, but I wasn't doing aging research. I was doing cancer research. But, I'm a big believer in tumor microenvironment, right? So, and I think microenvironment shapes who you are as a person as well. And so when I was at that institute, I would go to these talks, and some of them I would find excruciatingly boring because people were talking about aging and going up and down stairs and how your knees break down when you get older and blah, blah, blah. And I'm sure when I talked, they were just as bored by me. So, that's no reflection. I'm sure their research was great, but it wasn't my field. But as I got more and more exposed to it, I kept thinking, all of these things break down as we age. And we know that aging is the number one prognostic factor for cancer, but we don't really explore that. Nobody really explores that. And so we started to we were lucky because we worked in melanoma, which is an aggressive cancer of the skin. And so we were able to get skin samples from young people and older people and ask, what did the normal skin, how did that change? And what did those changes mean for tumor cells? And that's basically what we've been doing for the last 10 years. And it's been so fascinating.
You have done a lot of research on cancers, especially on melanoma metastasis. Could you please briefly explain to us what factors contribute to the development and progression of metastasis and its resistance to current therapeutics?
Sure. You said briefly, but that could take me the full hour to explain, but I'll do my best. So, something that we have found that's so interesting in melanoma is that it undergoes what we call phenotype switching. So you have tumor cells that will grow in the skin. And when they are growing rapidly, they tend to be less invasive. So they tend to not really leave the skin as effectively. But the tumors that grow more slowly actually tend to leave the tumor site more rapidly. And in our newest study, what we found was something that I remember I mentioned that it was, data that we found that kind of turned our previous data on its head. So we had been looking at sort of this yin and yang of growing or going. So if a tumor cell was growing, then it wasn't going. If it was going, then it wasn't growing. And that was regulated by a family of proteins that we call the Wnt proteins. And different Wnt factors did different things. So some Wnt factors drove the growing phenotype, and some Wnt factors drove the going phenotype. And so we were super interested in this particular factor called Wnt5a that we know to be critically important for melanoma invasion and dissemination from the primary site, so where the cells leave the primary tumor. And but what we weren't prepared for that we discovered, which was, both was both confusing and then very exciting, was that the cells that had that high Wnt5a were the ones that left the primary tumor on the skin and went to the lungs. But when they got to the lungs, they went to sleep. So this is something that we call tumor dormancy. The cells don't grow, they don't divide, they just kind of hang out there. They stay for a really long time. And it was the cells that had the high Wnt5a, which we had always associated with worse outcome and more metastasis. But it was the cells that had the high Wnt5a that were the ones that didn't grow in the lung and that colonized the lung. But as the lung aged, there were factors that began to change in the normal cells in the lungs, and the fibroblasts specifically, that when secreted, deactivated the Wnt5a, and suddenly those cells started to grow like crazy. And so it really explained to us a clinical observation that had been there, that has been sort of a paradigm of cancer for a very long time, which is that you can remove a tumor or you can treat a tumor. And then, within several months or sometimes several years or decades, the tumor will come. Like first you think the tumor is gone, everything is fine. And then later, when a patient is in their 60s or, 50s or 60s, that tumor comes back with a vengeance. And that's a process that we call tumor dormancy and emergence from dormancy. And so our work in this has shown that now as you age, something that happens normally during aging can drive those sleeping cancer cells out of their sleeping state and make them become an overt metastasis. So that has been super fascinating recently. Some of those same mechanisms also make those cells resist therapy. So Wnt5a, again, it drives survival of these cells, which allows them to survive in the lung, but it also means that it allows them to survive during therapy as well.
Well, Professor, cancers are more prevalent among elderly people when compared to younger ones. Is there any connection between aging and tumor progression?
We like to think so in our lab. So we know that when we take normal cells from young and aged people, like skin fibroblasts, for example, and we examine the things that they secrete, they behave very differently. And if you take a tumor, the same tumor cells with the same genetic changes, and you put it in a young mouse or an old mouse, those tumor cells behave very differently. They don't grow as fast, but they metastasize more, and they have more angiogenesis, and they're more aggressive, and they resist therapy more in the elderly. So there's a lot of molecular connections between the way we age normally and what we see those aging cells can do to cancer and to tumor cells.
Most people think cancers are inevitable. Is it true, Professor?
So I unfortunately agree with that view. I do think most cancers are inevitable as we grow older. As humans, we weren't really designed to live this long, right? Our Neanderthal ancestors, they probably died in their 30s and 40s. And so now with antibiotics and other treatments, we're living 70s, 80s. And so our cells tend to break down. The DNA repair systems go wrong. And so I do think that cells mutating and becoming cancer cells, it's inevitable. But I don't think dying from cancer is inevitable. And I think that's something a mindset that we're really going to see changing dramatically over the next several years. Already, President Biden in his moonshot declaration, for example, said we're going to learn how to manage cancer. So people always used to talk about curing cancer. And I have always thought that it's very important instead to learn how to manage cancer the way we manage something like diabetes or high blood pressure, right? So you can have a disease, but if you're treating it appropriately, it's not going to kill you, or at least not right away. So.
Professor, you mean that the connection between the aging and the cancer development is loss of cell function and changing the DNA genetic composition, and I found and I watched in one video that the losing of fibroblast when we age allow to get spreading of the cancers more in the skin.
Yes. So well, I think there's multiple reasons. So there's changes in immunity. And I will also say that when we are looking at all of these things, we're realizing more and more that it's very organ specific. And so each organ ages differently. So for example, in your skin, the fibroblasts in your skin, they tend not to renew and turn over the way fibroblasts in other organs, like your intestine, for example, will renew and turn over. And so, we see changes, as you mentioned, in the DNA, so genetic accumulation of genetic damage. We see changes in the factors, the growth factors that these fibroblasts secrete. We see changes in things like lipids and exosomes. So all of these different secretory factors, the metabolism of the fibroblast changes, and that drives, differences again in the things that they secrete or the way they use the oxygen. And in addition to all of that, we also see changes in things like the immune microenvironment of an organ or a tumor.
Okay, that means
very complex.
That means the mutation in certain genes induce different type of cancers, like lung cancers and prostate cancers. Also, there are two different type of cancers. So the certain mutation should be come to the particular gene, isn't it?
Yes, but I think that you can have tumor cells that are initiated. So for example, in melanoma, the BRAF oncogene is a big driver of melanoma, but you find plenty of normal lesions that might have that mutation, but have not yet become cancer. And so what we think is happening is that although some things are genetically driven, the changes in the microenvironment around them allow them to, what's the right word, hijack that mutation and use that and become more aggressive, if that makes sense.
Okay. Professor, recently I have read in some news that drug in clinical trials, which is called dostarlimab, did a miracle. I mean, cancers in all patients vanished for the first time when treating the rectal cancer. So do you think that will it effective again all type of cancers?
So I think the answer to that, unfortunately, is no, because it's only effective even against only one type of rectal cancer, right? So the rectal cancer that it's effective against has to have a mutation in MSH1 or 2. So these are, genes that are involved in genomic instability. And it's and so this is part of research that has been going on for quite some time. Actually, some of it originated at Johns Hopkins with Dr. Bert Vogelstein and Ken Kinsler. And Luis Diaz, who's the senior author on the paper you're talking about, was with them. And then he went to Memorial Sloan Kettering. And what they found was something very interesting, which is that lung cancers that bear that mutation are more sensitive to inhibition by checkpoint inhibitors. So the therapy you're talking about targets an immune checkpoint called PD-1. And I don't know how much you guys know about immune checkpoints. Should I say what they are really quickly?
Yes, sure.
Okay, so when you have a cell that presents some of these ligands that are called, so PD-1, CTLA-4, these are all immune ligands. These are all ligands that are presented on the surface of tumor cells that act to sort of dampen the immune response, right? Because a tumor cell wants to hide from it. And so when these antigens are present, the tumor cell tends to be less visible in a way to the immune system. And so if you inhibit those checkpoints, you make the tumor cell visible to the immune system can attack it. And so in melanoma, we have had a lot of success with these anti-PD-1 drugs, like the rectal cancer one. I can never say the names. I think they just make them purposefully difficult. But anyway, so that's an anti-PD-1 therapy. And so in melanoma, we're very fortunate because melanoma is one of those tumors that responds beautifully to anti-PD-1. And for the first time, we're able to almost, I use the word cure hesitantly, but almost cure about a third of all patients in melanoma, which is, fantastic. So again, though, a lot of melanomas express PD-1, but not the checkpoint inhibitors don't work in all of them. And so I think that we need to keep exploring those avenues because it's the first time that we see such beautiful remission and regression, but we'll need more and more of these types of drugs to really make an effect.
Thank you for making it clear.
Of course.
Professor, may I know the current research you are exploring and what remains to be explained in the field of cancer?
Oh my goodness. So I can't answer the last one because it's too much. There's so much that we need to think about. So, my lab continues to focus on understanding the intersection between aging and cancer, because if you think about it, in a lot of the studies that are published, right, we publish them on mice that are age 6 weeks old or 8 weeks old, or we use the younger, healthier patients in our clinical trials. And the truth is the patients who are getting cancers are really in their, late 50s, 60s, and those are the ones that we should be modeling in mice. So we only use 18-month-old or older mice, year-old to 18-month-old. And, we're really focused on when we see a finding, what does that look like in young patients? What does that look like in aged patients? We're now also examining differences between male and female patients. So how do these different observations hold true in patients who are women, men, young, old? And it's been really fascinating, some of those new data that are coming out. We're also continuing to, we had an observation a couple of years ago that older fibroblasts accumulate a lot of fat, like many of us do when we get old. And then they secrete those fat and the tumor cells take them up and they use them to resist therapy. And if we can inhibit that uptake of those fats, we can make those cells sensitive to some of the therapies we have available. And those results were very dramatic. And so really what I'd like to do over the next couple of years is to finish validating the inhibitors that we have and get those into clinical trials.
I'd like to learn more about your outreach activities. How do you reach out to students and the general public outside of your lab or university?
I didn't hear all of that, but I think you asked me about my outreach activities.
Yeah.
Okay.
How do you reach out to?
Sorry. So I do a lot of things on social media like Twitter, Facebook, Instagram. And I do a lot of, again, with melanoma, I'm always cautioning people not to go out in the sun and get burned, to avoid tanning beds, to wear their sunblock. So in the summer, it's very lonely for me because most of my friends just avoid me all summer because they don't want to get yelled at if they have a tan. So, but yeah, so I do a lot of stuff on social media. I also do a lot of grassroots activity, Hopkins is in Baltimore. And so we have a lot of schools that have a lot of underrepresented minorities in the school. And so I really like to go to elementary schools, middle schools, high schools, talk to the kids, tell them about science. I feel it's very important that they see someone, who looks a little bit like them, being a scientist, and that they too can know that they can go on and be a scientist too. And so I'm a big believer in representation mattering. And so I like to do that. So that's sort of a very grassroots thing. And as you can imagine, I only get to do that a couple of times a year. Through the department and through the school, we reach out to the community a lot. One of the things that I've done in the past that I'm hoping to do in Baltimore too is to, when we go to sort of a baseball game, it's a big stadium and we set up these booths at the front of the stadium and we hand out information about sun protection awareness. We hand out little packets of sunscreen and tell people not to forget to put it on during the game. And so that's been really interesting because I've had people reach out to me after the game and say, oh, I never thought about the fact that I have to reapply my sunscreen, or get my moles checked. And I went to the doctor and they found this mole and it was a precancer. So thank you, because I would have ignored it, So I joke a lot that I do more for melanoma research on social media than I do in the lab, because I don't know how many times I've posted something and someone will be like, oh, I saw your post and then I noticed this. So I'll give you a concrete example. So a couple of years ago, I was getting a pedicure and I looked down on my toe and I saw a line on my toenail, and it literally looked like someone had taken a felt tip pen and drawn a straight black line down the middle of my toenail. And when I saw that, I panicked because that looked like what we know to be a form of melanoma called acromelanoma. It's actually the melanoma that Bob Marley, if you guys know Bob Marley, the famous reggae singer, that's what he died of. And so when I saw that, I have to say I panicked, right? And of course, I called my friend, the dermatologist, and I was like, Chris, you have to cut my toe off. And he's like, all right, just come in, let's look at it. And so I went and he took the whole toenail, and it was a pre-malignant lesion. So that was good because it wasn't cancer yet. But I posted this story on my Facebook page because I just wanted people to be aware, because I was like, I knew what that looked like. But anyone else might have just ignored it and been like, oh, it's a weird toe fungus or something, right? And so I posted a picture and I posted the story on my Facebook page. And then a friend of mine contacted me, a friend of mine from Africa, actually, she contacted me and she said, I saw your story and I had noticed something like this on my husband's toenail. So I immediately took him to the doctor. And sure enough, he was a stage 1 melanoma. And so I feel like, I do so much more in a way by bringing people's attention, making them look at their moles, making them go to the doctor, making them wear their sunblock than I ever do in the lab, which, it's a little disappointing in one way, but it's also good in another way.
To students who think that doing research and balancing life is tough, do you have any advice or suggestions?
I always say just jump in with both feet because, at some point, I have to say my life is completely insane right now. I live in 2 different cities. I have a 17-year-old daughter and a husband. And I am doing I chair a department. I lead a program in the cancer center. I run my lab. So things are crazy, but I love it all and I embrace it all. People talk a lot about work-life balance. It's very important to just find those things you love and really make sure. And I'm going to be a complete hypocrite. I am fully, my lab, I tell them all the time, take your time with your family. You don't have, I try to encourage them not to work weekends if they don't have to. And I always tell them take time for self-care. I'm a complete hypocrite because I never do that, but that's how I like to. I like living a mile a minute. And then there are times when I realize, things are. There are times where I'm like, okay, I need to step back and take a break because, for example, I was in Baltimore, which is where Hopkins is and where I work. And my husband was in Philly and he texted me and he said, there's a basket of clothes in the kitchen. Are they dirty or are they clean? And I said, I don't know, smell them. And he wrote back and said, ew. And so then I was like, yeah, I think things have gone off the rails a bit this week. Maybe I'll calm down next week and cancel a couple of meetings and try to get my house back in order. I like often young women will say to me, oh, I want to have a family. Should I wait? When you're ready, just do those things. And if you're in an environment that's not supporting that, then find a new environment because that's, that you should always be in an environment that supports who you are as a person and your life outside the lab as well as in the lab.
Well, Professor, I am interested to know how you spend your leisure time outside the lab and how do you manage your time?
Yeah, so I just told you I don't manage my time. Not well. No, that's not true. So I'm a big believer in lists. So I always assign myself like critical tasks that have to be done each day. In terms of managing my time, I just embrace it all. I do what I have to do when I have to do it. I'm pretty organized, which I think is a good thing. And that really helps me with my time management skills. For leisure time, what I love to, I'm very social. I guess that's a Sri Lankan in me, right? So we love to always have people around and always have people over. I love to cook. And so for me, cooking is really my stress relief. And, I think there's rarely a weekend day where we don't have people over. So the pandemic was very hard for me because it meant a lot less social life and a lot less people coming over. But
Well, Professor, as an eminent scientist, what advice would you give to the younger scientists like us who are willing to enter the field of scientific research? And what advice would you give us when choosing a certain area in science?
So that's a great question. So, I will say that the one thing you have to be if you're going to be a successful scientist is persistent. You have to believe in yourself, and you have to, a lot of people along the way have always told me, I can't do this, or I can't do that, or I should not get a PhD and I should go get married, or if I, so on and so forth. Like there's been a million. I was told, don't bother going to grad school. I was told, oh, you should just, don't bother becoming a PI. You should just take care of your kid and get married. Get married and take care of your kid. And so, if you feel like this is what you want to do, then don't give up on yourself. Just make sure you pursue your dream. Don't listen to all those voices in your head who are trying to get inside your head and tell you not to do something that you firmly believe you can do and that you want to do. Because if you have faith in yourself, you will find a path. And so that's my advice to you is don't give up. And there will be times where it's so hard and you think, what the heck have I done? And why am I doing this? And if it is wrong for you later down the line, then it's wrong for you, right? But I think that letting other people tell you it's wrong for you is not the way to go. If it truly is wrong for you, then you will learn that on your own, but don't give up until you get to that point. So perseverance is absolutely critical and very important. In choosing a field of science, go with what you love, go with what fascinates you. I just told you guys that I'm just as fascinated by a dirty pond as I am by cancer research. But for me, cancer research was a personal thing. That's, why I became so interested in it. And in a way, when I started out, I wasn't interested in looking at melanoma. And I became interested in it because of the science and because of the project. So always follow the science. But when you get to graduate school and you're looking for a lab, look for the labs that are labs that take care of each other, that collaborate with each other, where the students are happy and where the students, want to work together, where the students actually like each other. Those are all very important things. And make sure that any lab environment that you enter is a lab environment that is not abusive, but that is a lab environment that supports you and lifts you up and helps you get to your goals. That's so important. Sometimes, you'll be tempted to go to some big name lab where everyone's like, oh, that's, so-and-so, and they've discovered such and such. And, they get Nature, Cell, and Science papers all the time. And you might be tempted to go there, but it could be a miserable environment, or it could be a great environment. So, all I'm saying is that again, I'm a big believer in microenvironments. So, make sure you're in a microenvironment that supports your growth and development as a scientist.
It's a great advice, Professor, because especially for me, because I am afraid that if I'm going PhD and I have to work hard and have to publish articles. So if I get married or kids, then I will don't have the time and spend the time with family. I'm afraid, but you make it clear.
I think if you want to have it all, you can have it all. And you will be tired. I'm not lying to you. When my daughter was little, I was very, very tired. But, it's fully worth it. I don't regret a second of it. I don't regret the sleepless nights. I don't regret having her for sure. And I don't regret my career at all. So just have faith in yourself and know that you can do it. And it's very important to have a supportive partner too. I'm very lucky that way. My husband is an equal partner in our marriage, and that's super helpful.
It's good.
Even if he won't smell the dirty clothes, he's still an equal partner.
Professor, you said we have to have the good microenvironment even in our lab. So what do you think what do you think is necessary to be a good mentor to young scientists? Definitely they should be family friendly.
Yeah. So I think what's really important to be a good mentor to young scientists is to be able to put them and their development ahead of your own needs and your own ambitions, right? And by that, I mean, sometimes people think of, there's sort of a perception. I don't know how true that is anymore, but it certainly was true when I was going through. I was very lucky. I had amazing mentors. So I'm not speaking from personal experience, but I knew a lot of people who were in lab environments that were abusive, and they were told, like, if you don't work till midnight, I'm going to fire you, and your visa is going to, expire, and blah, blah, blah. Right. So I think and sometimes people would do that because they felt like they needed someone to work hard and do the experiments so that they could publish the papers and they could get the next grants. And so that is important, obviously, right? You want a lab group that will work hard so that the work can be done and grants can be gotten and papers can be published. But at the same time, it's very important to, each person is different. And that's what I've learned as a mentor along the way. And when someone comes into my lab, the first thing I say to them, especially postdocs, is, where do you see yourself? What is it that you think you want to do? And you're not bound to it. Like, you can change your mind, right? You can do this for a year and say, no, that's not what I want to do. And then I'll help you find the next thing. So really, helping your students and your postdocs put their own needs, help putting their needs ahead of your own, I think, is one of the most critical aspects of being a mentor. For me, my greatest successes is seeing the people that I've trained succeed themselves. That's what I would say I think mentorship is.
Yes, I also would like to have the mentor like you.
You might want to talk to my students first, but
last but not least, could you please give us a comment about this interview?
Yes, it was a lot of fun talking to you guys. It's so lovely to be, I have to tell you that recently, for whatever reason, I was going to say, I wish this could have been something we could have done in person. And partly because the last several weeks, I have really been craving a mango that actually tastes good, because I have not had a good mango since I was last in Sri Lanka, which was 2019. So I really miss the mangoes. But you guys are wonderful. You asked such great questions, and you're all so nice. And I'm very excited about the future of Sri Lankan science, listening to all of you.